Okay, everybody, this is going to be our lecture on the properties and functions of biological macromolecules. To give you a little bit of a heads up, this is actually two sections that we're going to roll into one. It makes a lot more sense to kind of do them all at the same time. 1.4 is all about what they look like, while 1.5 is kind of more so of what it is that they do. Let's go ahead and get rid of that. So we go and take a look. It says the structure and function of polymers are derived from the way their monomers are assembled. If you remember from a previous lesson, right, we talked about how molecules have specific behaviors depending on how those atoms are arranged with each other. Unequal sharing of electrons, some areas will be partially positive, some areas will be partially negative, and that's going to determine how it behaves around other molecules, whether they're attracted or repelled or combining with other ones, right? In the case here, we're looking at how the structure or how we line up these biological macromolecules and how it actually ends up changing their specific function. Now, our four types of macromolecules we'll be going over are nucleic acids, proteins, carbohydrates, and lipids. What we'll do is that we're going to go over the monomers and then talk a little bit about the polymers within these and just give you some structural information about it. In the case of a nucleotide, it is made up of a phosphate group, a nitrogen base, and a sugar. Okay, so I'm going to go ahead and start doing that. So the phosphate group is going to be this guy right here. So it's very, very negatively charged, lots of oxygens. We know it's a super electronegative element located over there, especially with phosphorus in there as well. It's a pretty wild section of this. The next one is a nitrogenous base. The nitrogenous base is located over here. There are 5 different bases that are out there that will give you the 5 different nucleotides, your A, T, G, C, right? If you think back to them, your adenine, guanine, cytosine, thymine, as well as uracil, that's going to be from the nitrogenous bases. And finally, a pentose sugar, that's either ribose for RNA or deoxyribose for DNA. And that sugar is located right here. To be able to identify which one is which, we're going to go and take a look at this little section of the molecule right down here. We'll see that it's either an H or an OH, and that will be able to tell us if it is deoxyribose or ribose. Now, the overall purpose of nucleic acids is that they encode biological information in a sequence of nucleotides, right? So if you remember from your time in biology or biotech, you know that you would have a specific order of a specific order of nucleotides in your DNA, and this would end up basically be coded as codons, and we'd be able to turn this into mRNA and then make a protein out of it, right? In the case here, this is what we mean by it's encoding information. It can either encode it as DNA or as RNA. Here, we're just seeing the different areas on the different nucleotides or the nitrogenous bases that are associated with our nucleic acid. So we said adenine, thymine, guanine, and cytosine. That's specifically for DNA. And they're going to go into more detail about RNA. It's the last section that's in here. I kind of find it weird that it's its own little section, but I'm not going to worry too much about it. Adenine, guanine, cytosine, and uracil, that's going to be for RNA. So in the case here, they have the U while DNA has the T. We can end up seeing that we end up combining the base and the sugar, the pentose sugar, that's going to be making the nucleoside. And then once you end up adding the phosphate to it, that ends up making it the nucleotide. The way that we combine all these guys together is called a phosphodiester bond. Certain ones of these molecules will have specific terms that we use in order to describe how they're actually linked together. So phosphodiester bonds are specifically with nucleic acids only. Nucleic acids. So I may want to write that down because if you end up saying like a polypeptide bond or a glyco linkage or something, you actually be using the wrong verbiage. So phosphodiester bonds are specifically for our nucleic acids. If we end up seeing this right here, this is showing us a picture of a nucleic acid chain, a segment of DNA that is starting to be built. It's a polymer, right? So we're now making a polymer of DNA. We're going to go, and I just kind of want to make you aware of this 5 to 3 prime. This is very important to understand the directionality of DNA and how things are done. Everything is done 5 to 3. So we can see that it's attached 5 to 3. It's attached 5 to 3. And where the name comes from is specifically where this carbon's located. So it's either the first, the second, the third, the fourth, or the fifth. And that's actually where the name 5 prime, 3 prime ends up coming from. What also I want to draw your attention to, right, is a dehydration synthesis reaction, right, where we take 2 hydrogens and 1 oxygen, and we're able to create a molecule of water, and we actually link them together. So what happens is that we remove these 2 guys, and then we're able to use a carbon right here and combine it to that oxygen right there, as we see right here into that spot right there. This is the phosphodiester bonds that we were taking a look at, right, the ones they end up talking about right here. Your next group is going to be your amino acids. So we are now moving into amino acids, right? This is your proteins. Okay, these are your proteins. Your amino acids are the monomers. The polymer is going to be what's called a polypeptide. We'll show you that in a little bit. Okay, it's a little bit strange because it says everything but storage, energy, and genetic information. Wait, that means everything else? Look, if it does something or does like a function, has a function, that is going to be these proteins right here. They basically are the movers and shakers of your body, right? Genetic information is all nucleic acids, and then energy and membranes is those carbohydrates and lipids. If it's something like an antibody, if it is a channel within your cells, is it a I don't know, an enzyme to help out with chemical reactions. These are all proteins. They basically do everything. Like if you think it has a job and it's actively doing a job in your body, it's a protein. The structure of this monomer is made up of 3 components. I guess you can maybe argue a 4th one, but we don't really want to go too far into it. You got a carboxyl group. Okay, that carboxyl group is this guy right here. For your situational awareness, there should be some more hydrogens or oxygens in here. This picture is a little misleading. I'm just going to kind of roll with it right now. Okay, this is the carboxyl group located over here. We can tell because there's like a carbon located over onto that side. Your next component we go and take a look at is going to go ahead and be the amino group. I kind of messed over that one. The amino group or amine group. That's going to be this guy right here. The amino group is where the name amino acid comes from, and it's where this nitrogen, specifically this nitrogen with the hydrogens attached to it, is what ends up getting that amino acid. If we end up going to take a look here, the last component to it is this R group, right? So if we go and take a look, that R, right, that R is just a placeholder. Okay, it's a placeholder. Reason why is that there are 20 different placeholders that we can end up putting right there. Okay, and they all are a different amino acid. They do something a little bit different. Okay, overall, it still is an amino acid, but how they are oriented, right? If we have an orientation that looks like this, right, and we have like amino acids 1, 2, 3, 4, 5, but then I take that same numbers, but I do like 1, 3, 5, 4, 2. These amino acids, this amino acid sequence will do something completely different. And it's all about how these side chains actually interact with each other, which we'll kind of discuss a little bit. Okay, this right here shows you the image of all the different R groups for the amino acids, right? There's 20 of them total. Now they're grouped together by like chemical properties. All right, it's like chemical properties here. What do we mean by that, right? Well, chemistry is the study of life, right? I want you to think about their intermolecular forces. That's probably an easier way to think about how these guys are grouped together. Now, if we go and take a look at these ones right here, we have an electrical charge hydrophilic chain. right? So we have some sides that are positive and some sides that are negative. What that means is that in an amino acid sequence, this arginine right over here might be attracted to this aspartic acid that's located right there because their opposite sides are going to be attracted to each other, right? It's like that hydrogen bonding that we were talking about earlier, right? It's those IMFs, right, that we're discussing. Another group of these intermolecular forces that are unique to our proteins are these polar hydrophilic and these hydrophobic interactions. I want to go do this. So here's our hydrophilic, which means that they are water loving, right? Water loving. So these side chains are going to be attracted to water. So if my protein looks like this, right, and there's water on the outside of it, these specific side chains are going to be attracted to the water and it's going to cause the protein to twist in a specific way so they're facing it. Opposite to that, if we go and take a look at the other one, this hydrophobic, right? This is hydrophobic side chains. These guys are water hating. Water hating. Which means that if there's water on the outside, right? Well, these guys don't want to be near the water, so they're going to be facing inwards. So they're not going to be facing that water. So it's going to cause the protein to twist in a specific way. So then that way, these ones are not facing the water, right? And now you can kind of start to imagine, right? This is going to start to develop a three-dimensional shape, which we'll talk about in proteins in a little while. But what that does is that because the amino acid sequence is in a specific order, it's going to fold in a specific way and it's going to have a particular property that's associated with it. You got these other special cases. I'm not going to do special color. They just kind of do some really weird, funky things. Specifically, this guy right here, the sulfur will make these things called sulfide bridges. We'll show you them in a little while. Just like how we ended up showing you in the nucleic acid example, look at this. This is another dehydration synthesis. I take two amino acids, right? I'll take an oxygen, I'll take some hydrogens. I'm going to go ahead and remove H2O, right? I'm going to end up removing those. And then sure enough, I end up combining them together and I'm starting to build what's called a polypeptide, right? Polypeptide right there. Now, this specific thing is called a peptide bond, right? Not a phosphodiester bond. We're not peptide bonds. We need to learn about all these guys. Yeah, I know. The nuances, the details are so fine right here. So you just need to maybe do like a flashcard or something. I'd almost maybe say like a chart with all this information, the type of bonds would definitely be helpful for reviewing and studying this stuff. This is just showing you right here as we start to build our amino acid sequence, look at the different side chains, notice how they all are different from each other. They're just going to want to form in a specific way, which makes them very, very unique. That will make it have a very interesting three-dimensional form. So one time it might twist like this. If I take those same amino acids, it might twist in a completely different way because that's just what it's going to go and do. One thing I also want to make in mind is that if you remember back from those elements of life, we talked about sulfur. This is where sulfur shows up very commonly, but this is why I said that not all amino acids have it. So you could make an argument that sulfur is found in amino acids, right? But just not in all of them. Just kind of making you aware of that. All right, we're moving on into carbohydrates, the delicious ones, right? So in the case here, carbohydrates, we're going to go talk a little bit about verbiage. Okay, carbohydrate. What I want you to do is take a look at this deoxyribose or this ribose, right? And well, we'll go take a look at this guy, right? And I want you to go and count up the number of carbons, hydrogens, and oxygens in each specific one. How many carbons, hydrogens, and oxygens do you find in each specific one? Okay, so giving a little bit of time, you should have made a little bit of a count. And I believe that in this specific scenario, you're going to end up having about like 5 carbons. You're going to have somewhere around 12 of these hydrogens, and you should have, what is this? Looks like 3, 4, looks like 4 of these oxygens. Okay, so these ones are a little bit tricky to go do because ribose and deoxyribose, these pentose sugars, don't really normally play by the normal rules, right? Now, what I'd like to go do is actually do this with the 6 carbons, and this is going to be a little bit better to work with. Specifically, let's go with glucose, good old-fashioned glucose. Do the same thing. Give me the C, give me the carbons, the hydrogens, and the oxygens. Just count them up real fast. Hopefully, if you counted, you should end up finding this thing where it said C6, we have H12, and then O6, right? This is the chemical formula for our glucose, right? Now, if we go and take a look, you'll see it's actually the same thing on all these guys over here as well. You end up taking a look, you'll see like, oh, they have the same amount of carbons, the same amount of oxygens and hydrogens and all that other fun jazz, right? What you're noticing is like what makes them different is like, oh, the OH is down here as opposed to the OH being up there, right? Remember, the structure is all about the function. So because that, even though they're made of the same stuff, they look slightly different from this one little tiny minor detail. That's going to make it so that they have different functionalities or what can actually interact with it. Now let's go back to the term carbohydrate, right? Well, think about the word hydrate for a second, right? If you're hydrated, what do you have? Water, and water is H2O. How many times can you make water out of that H12O6? Should be 6, right? You got 12 hydrogens, you divide that by 2, you can get 6. You get 6 oxygens, divide that by 6, you get the 1. So H2O. So carbohydrate literally means that for every carbon, you're going to have 1 water. That's actually where like the name kind of comes from. Now, it kind of breaks down if you look at these scenarios right here with the pentose sugars, but overall with carbohydrates, this is the method on why they actually have the names that they do. Now, let's talk a little bit about them. They are called monosaccharides. So a monosaccharide is an individual carbohydrate. So it's going to be found as these rings. So that's a monosaccharide, that's a monosaccharide. All these guys are monosaccharides. And they're used for short-term energy storage, right? There's a lot of energy that's located within them. And their structure is that they are a ring structure, right, with anywhere between 5 or 6 carbons. They form a ring. Now, when we start talking about cholesterol, you're going to be like, oh, look, they're rings. Or let's look at the nitrogen bases, right? And I'll actually show the nitrogen bases for a second, right? This is something where students get confused with. I kind of want to make you aware of this, okay? This is a carbohydrate. That's a carbohydrate. This is a carbohydrate. These are not carbohydrates, okay? The carbohydrate will have that specific 1 to 2 to 1 ratio associated with them. Those nitrogen bases are not. When we start showing you cholesterol, you're really going to kind of see that. So we'll see that in a little bit. Now, when you start to combine your monomers, your monosaccharides together, we start to make these polysaccharides. And all what's happening is that, hey, look, it's another dehydration synthesis reaction. Go figure. That's actually how all these things happens. We make a linkages. Oh my gosh, I can't speak. A linkage, and what that's going to end up doing is that it's going to go and allow us to combine these two monomers together. And what's really neat about it is that when you combine those monomers, you can start to chain them together to make some really freaking cool stuff. So in the case here, we look at starch, right? This is going to be what was found like in the banana lab that we did, right? That banana or the potato, right? It's just long lanes of carbohydrate that just give it a very, very stiff look. Versus in the case of other cases, we look at glycogen, it's highly branched, right? Lots and lots of branches. We can see it branches here, here. And like it's like there's so many branches. Well, the reason why is that glycogen is used in order to basically cleave it off. So it's like, okay, I need to get a glucose here. I'm going to get glucose here, glucose here, glucose here, glucose here, glucose here, glucose here, glucose here. And you're able to lob off all those glucoses in order to go and use for your cells in the case of your blood sugar is low. But if you go and take a look at starch, Christ, man, you have to go somewhere, right? We don't even see the start of the chains anywhere near here. And we have to go and figure out where the end of it is, right? So, at least in the case of the banana lab, what's happening is that the starch is breaking down and eventually breaks down, and then more of it breaks down. And this is why it actually gets all moldy and not moldy, but gets all ripe and stuff because it eventually all starts to break down together. So it's almost like it's almost like an exponential. It's almost like an exponential when it comes to ripening. Like it gets a little ripe, and then it's like it's a brown banana at that point. And you don't want to ever eat it unless if you're doing baking. Okay, so there's your carbohydrates. Here's about lipids. Lipids are weird. They're fats, okay? Making you aware, the monomer is a fatty acid, but I'm going to do a little caveat to this, okay? They don't combine fatty acids and they don't make, their polymers are weird. The polymer is weird here. The reason why is that there's not really one. What we can do is that I can take this glycerol molecule and attach 3 fatty acids to right here, and I can end up making a triglyceride. Uh, Glyceride, right? I can end up making a triglyceride. What's kind of neat about it is that, hey, cool, look, I did it. Like it ends up combining with your H2O-ness, right? Like how anything else, because you have like these areas that you're able to attach to. Sorry, I got some in my throat. Excuse me. All right. It attaches to itself and does dehydration synthesis like normal. The difference is that when it comes to your fat cells and stuff, all you're doing is you're just making more and more and more of these triglycerides. And that's actually what makes fat cells really, really big. And when it comes down to, geez, guys, I'm so sorry. I need to get like a drink of water. Like I am, something is in the back of my throat. It is like the worst. This is my second time doing this recording. I'm not recording this again. You hear my frustration in the background. All right, there it is. All right, feels a lot better. So you end up making more and more and more of these triglycerides. We can end up increasing the length of these fatty acids. That's actually what causes fat cells to shrink. You know, when people say I'm burning body fat, you're not actually destroying the cells. You're just making them smaller. It's kind of fascinating how that ends up going. One thing to kind of keep in mind when it comes down to your fats is that you have 2 different kind of groups of them. You have your saturated and unsaturated. Your saturated is your yummy, delicious butter. Basically, what it means is that it is a solid at room temperature. The reason why is that we have all these single bonds right here, and we're able to keep all these triglycerides, all these fatty acids close with each other, and it's perfectly fine, right? Now, the other type that you have is going to be what's called a unsaturated fat. We'll do this in blue, right? Unsaturated fat is due to a double bond between the carbons. What does that mean? That means that instead of me doing single bonds, at certain points, I'm going to have a double bond. And what that does is that it actually changes the angle in which the elements are bonding or the atoms are bounded with each other. It actually forms a kink. And it can end up forming these kinks in the molecule that makes it so you can't really stack them close with each other. You know, you imagine that if you are like at a rock concert, right? And here you go, you're trying to pack people all the way close, right? And like, all right, cool, we got a lot of people. It's like sardines. Well, this is like going to a concert, but like the person next to you is like, I don't want you to be anywhere near me. Like, do not touch me. And you can't even get anywhere close to them, right? That's what's basically happening here is that the molecule is kicking. It's making distance between it and the neighboring molecule, so that you have all this open space right here, and that's causing it to be a liquid at room temperature. Now, if you end up heating butter, right? We know we heat butter, it melts, and you can go and eat that delicious butter. Oils are, they're always like at a liquid. It's just basically saying it's like, are you liquid at room temperature or are you a solid at room temperature? Right. The last one to go talk about is our phospholipids. These are super important. Okay, so the purpose of lipids is that they do long-term energy storage. They can also be used as membranes, right? Membranes. Specifically, this is a membrane that you can end up finding within the cell or any organelle. Right, it's very important, right? The cell and organelles are all made out of these membranes, right? And it's basically all these phospholipids. So instead of us doing a triglyceride like before, I'm going to go and get rid of that. I'm going to go and stick a little phospholipid head on there, and boom, that's basically what we just did right here. What this is that this is a super hydrophilic head, and you got these hydrophobic, right? The hydrophobic tails, which makes it so that it orients itself so the water, like the heads face the water. They love the water. They're like, we love water, right? But they almost like they don't want to get their feet wet. So what they do is that they orient themselves so then that way all the tails are facing away from each other, so that we can actually make a barrier from that, and it's pretty cool. All right, that's going to go ahead and stop this video. Hopefully I don't have to re-record this, and we will go and do 1.5 next.